Thank you very much. Thank you. So my basis, I'm a biologist, I'm not a medic, and yes, interdisciplinary, yes, application, stem cell biology. And my interest has been translating the things that we do in the lab together with Dieter into the clinic. I've taken a bit of a step back. If the theme today is cell therapies on the horizon, my job is to talk to you about production. And I'm going to concentrate on the ATMPs. So a lot of what we've seen so far, the stem cell transplants, the T cells, these are standard cell and tissue therapies out of a regulatory viewpoint. And I'm going to concentrate a bit more on the things that are moving, so the changing regulations in the field of advanced therapeutic medical products. You've heard about MSCs, everybody's talking about CAR T cells. These are classic examples of ATMPs. And I hope in 20 minutes' time, if not shorter if I can manage it, you will understand at the end where we are and what the things are that you will be dealing with. Okay, so I'd like to talk firstly about the origins and the types of ATMP, and then how they are progressed from the basic good idea into a clinical application. We've seen already how long that can take. I'm going to spend some time talking about this good manufacturing practice that everybody's heard of, and most people know what a lab looks like, but very few people know what really lies behind it. And then I'm going to talk about some new considerations that the EU have been having about this special case, because ATMPs throw up all sorts of problems if you really want to make your regulation so that people can do something, rather than making your regulation so nobody can do anything. Okay, where do ATPs come from? It used to be easy. So doctors used to have these toolkits, they used to have medicines, they used to be able to do transplants, or there were transplantable devices. This is effectively biology, chemistry and physics. And biology, chemistry and physics have merged over the years. So on the fronts of these applications, various things have appeared, and examples would be the CAR T cells. So we have cells that are actually genetically modified and turned in that, because of that, into medicines. We have devices that can be seeded with cells, or cell matrix devices. This is a cartilage transplant. And we have medical devices also that have nothing to do with cells but are coated with medicines, and those also come into a grey area. And these things exploded, really the first ones from the 90s onwards, I guess, with the first gene therapy attempts, and that was a very long time ago. They exploded for a while in a sort of unregulated fashion, and a lot of things were done that were not very well controlled, and a lot of things were done that were very, very exciting and very optimistic, and in the end not very successful, and the whole thing screams for regulation. So the EU began to get involved in this, and the ATMP regulation, the first one was 2007, a really decisive one. And this is a big document, but effectively what it's based on is saying you don't bring these things onto the market. And market doesn't mean selling. Market means distributing, really. But you don't distribute these things without some sort of official approval. And you don't get the approval until you've shown that it's a good quality, that it's safe, usually the most important thing, and that it's efficient, it does what it's supposed to do. And that's often very difficult for an exploratory therapy. To produce them properly, there has to be a good risk management system, and you have to be able to say 20 years further on what happened to this patient. So there has to be a long-term traceability. And to assure that this happens, there is a system of scientific assessment offered by committees in the European Medicines Agency, and those are primarily the CAT, the Committee for Advanced Therapies, and for some things also this Committee for Medicinal Products for Human Use, GMP. So what the CAT does is help decide. You can try and decide, if you like, yourselves, if you have an ATMP or not, but the CAT offers then this help, and is very often brought in to decide if something is an ATMP, if it's in this grey area, if it falls under the ATMP regulations, or if it's not anything particularly new and dodgy, so it can still be just a medicinal product, a tissue cell preparation, or a blood product, that are also regulated by more early and easier regulations. There was in 2014-15 a new reflection paper by CAT to say how should this categorization take place. And if you're referring to, if you're developing some sort of ATMP, then you can look at this effectively to follow flow diagrams, I'm going to show them, but there's too much detail to go through. What this effectively says is, based on your product, you can ask a few key questions and decide mostly yourself for most things, is this going to be an advanced therapeutic medicinal product? So do I have these options open to me and these restrictions, or is it not? And then I have other options and other restrictions. And this shows you the decision tree down to CAR T cells to demonstrate, firstly, everything with a recombinant nucleic acid in it comes into question. Vaccines are excluded, so vaccines are not ATMPs. If the intention is to do a genetic therapy, then it's included as a potential ATMP, and if the genetic content is the active ingredient, then more or less you're there. And the CAR T cells satisfy all that. The other two categories that we have, there are gene therapy medicinal products, there are tissue engineered products, as I've shown, and there are cell therapy medicinal products. And most of this is the other table that enables you to make this decision. Again, it's in the document, there's no point to go through it. The reason I'm showing it is because it has two decision points that are always the sticking points for cell therapies, and these are shown here. So the decision whether a cell therapy that's being developed is an ATMP or not depends really on two things. Has it been changed a lot? Is it substantially manipulated, as is always said? If it is substantially manipulated, then it's an ATMP. Purifying T cells is not. Depleting T cells is not. Stem cell preparations, anything to do with the cell purification, as long as you've got no exposure to some sort of drug or activating agent and no culture, is not substantially manipulated. These are examples of what is obviously substantial manipulation. Expansion in culture, so all the MSCs that we saw this morning, automatically ATMPs. Genetic modification, the CAR Ts, and anything where you change the genetic material is. RNA is not necessarily genetic, so a temporary RNA transfection can get you by here. You don't have to be an RTMP. Also, though, if you differentiate or activate them, these are all substantial manipulations, and your cell suddenly turns into an ATMP and you're in a different set of regulations. This is an interesting one. Homologous use has also been a source of a lot of confusion. If I'm taking a cell out of a donor and putting it back into somebody else to do the same thing as it did originally, then it's not necessarily an ATMP, as long as I've not manipulated it. If it's doing something different, then it is. But who decides what it was actually doing originally? And in the end, what happens is that if a cell is being put back into an obviously different environment, then it is assumed to be doing a different job. So, classic example, bone marrow cells into heart. That is then not the original intention. Mesenchymal stem cells, probably they're doing when you transplant them exactly what they did before. Maybe one of their normal functions is immune suppression. But they're an ATP anyway, because they're substantially manipulated. Okay, we fall into then these regulations. The ATMP regulation 2007 is the most important one, but depending on which groups you've got, which sort of product you're talking about, there are other regulations. We don't need to mention them here. I would like to move shortly into the problems now. Now we've got the case, special case ATMPs. Why are they a problem? Well, this is the way that all drugs have been developed over years. We know the different stages of the clinical trials. We have an idea, there are preclinical experiments. Yes, in theory, we know that it's doing something. And then you go through a series of hurdles to say it's safe, small local trials. We have a dose finding study and showing that there's some efficacy. Normally, these are phase 1, phase 2 studies, and then a phase 3 study that's efficacy. And even later, you can go into phase 4 studies that accompany then use of the product. Those require then these authorizations. So you can have a clinical trials authorization for the whole of the clinical trials part. That's the responsibility of your country. So the EMA doesn't have to get involved in this. The different countries have different ways of organizing this, but in the end, they hold the responsibility for saying what you can do within a clinical trial authorization, whether you get it or not. And then there's the marketing authorization. It's a proven therapy. It can be distributed and it can be sold, and that's where the European Commission do get involved. The problem here is the cost. Well, at the beginning, the idea in the preclinical studies and animal experiments are fairly cheap. As soon as you go into producing things that you want to bring into a clinic, even in a small clinical trial, they need to be produced to certain standards. And here we're talking about the GMP standards. And as you go on to include more patients in more centres, insurance, then it gets a very expensive business. So one claim I read a couple of weeks ago said classic development of a drug up to €1 billion to get it from here to here. Takes about 15 years. It's a bit difficult when the patent protection is 16 years. So there's a huge investment for some of these things to get them to the end of the road, and it's a long time till the payback comes, which is why things like the CAR-Ts are now being introduced with very, very high price tags. That won't last. The system will have to change. But this is a problem even for normal medicines. It's a special problem for ATMPs because at the beginning, you've got a lot of unknowns. You're developing the drug as you go usually. And I wanted to jump shortly just to talk about GMP, because if you're talking about establishing production in this area, this is the bit that everybody knows. And this is what a GMP looks like. Unfortunately, what GMP really looks like is that. The lab itself is relatively easy, it can even be fun. What goes around it and what you need to maintain this system is an enormous structure that says, I'm sure about safety, I'm sure about quality, and I'm sure about the efficacy. And a lot of this is also written down in the ATMP guidelines, special ATMP guidelines for GMP, special GMP guidelines. It's not enough just to have a nice lab. You have to be able to organize your product flows and show that you can maintain particular levels of particle monitoring, particular levels of infectious agents in these so-called ABCD areas that we've heard about. A is classically here inside the hood. Then B, C, D are as a series of rooms where through a pressure drop you keep all the particles out. You have to maintain that and show that you've maintained it, the environmental monitoring constantly during production. The production that you do all has to be validated. You can't just say, I'm good, I think it works. You have to be able to show with repeated documented procedures that these processes all work. All the equipment needs to be qualified. That means it needs to be the right thing in the right place, and it needs to be maintained with us once a year. So your local authorities will depend what this is. There have to be systems that say if something goes wrong or if something's different, exactly what has gone wrong. I'm not going to carry on and go all the way around here, but you need to have in all these areas a very strong documentation and regulation system. The SOPs are probably that everybody's heard of, the standing operating protocol, so that describes more or less everything that happens in the system. And if we just move around here and come to the personnel, so your personnel have to be the right people with the right qualifications, they need to be continuously trained. I'd like to show you the organigram that we have in Leipzig, okay? To show you, to run the GMP lab, how many people are involved. Now, just wait before you sigh. Very few of these people are permanently involved in GMP. So I think it's safe to say that Dietke's department has managed to do this whole trick very, very efficiently. And one of the reasons is that most of us would like to do something else, and we're doing the GMP part-time, and we make it as efficient as possible because we do not like to spend too much time doing it. But you can see here, Dietke at the top as the qualified person. These heavy positions are requirements. So we definitely need to have these positions filled. And we have to have people who are responsible for the whole of the production area. We have different people for the production of the standard hemopoietic products and the MSCs, the ATMPs. And then underneath that, then you begin to get to the people who are really doing the work and the monitoring. We also have then the quality control system. This tends also to be split up a little bit into the two different applications, but the head of quality control with a deputy, all these positions have to have deputies, and then the people doing the work, which are sometimes internal and sometimes these are subcontracted. So I don't want to scare you away, but I did want to show you what GMP really looks like and also how big the organigram is. That doesn't mean that you cannot begin with a small concern. The last thing I want to do is put you off. But if we're talking about therapies on the horizon and we're talking about ATMPs, then these are the structures that you're going to have to begin to get used to. Okay, and we do want to talk about ATMPs and why are those such a specific problem compared with other medicinal products. Well, usually drug companies producing aspirin make enormous batches. If you're treating a patient, you're making one product, and that is officially a batch of drug product. So the amount of effort that goes in per batch is enormous in an ATMP project compared to a standard drug product. They're often very complex, the things that are being made. They're coming out of people, and people are all different. Look around. So the products start off, before you even start manipulating them, being heterogeneous and different. We also have extra safety issues. None of us knows exactly what we're infected with. Yeah? And similarly, we can check some things, but you don't really know what you're starting with. We're talking about fairly rare diseases. These aren't headaches. This is not aspirin. We're talking about rare diseases in a small market. So where's the interest for people with money to get involved in this? Where does pharma get involved? And for that reason, they're often developed in academic and not commercial settings. And now the EU have also said we acknowledge that. We see that academics are developing these things, and for that reason, we're going to be a bit less stringent, because we can see they're not going to be able to do the same sorts of things that a company can do. And in the end, very often, the mode of action is often uncertain. The things we've heard about today, CAR T cells, are fairly obvious what they do. Mesenchymal stem cells, the second biggest stem cell therapy after hemopoietic stem cells, enormous field. Nobody really knows what they do. So how do we check whether they're effective or not? How can we say whether a batch that's produced has a high efficacy or not? So all these things go into now a thought process by the EU that says we're going to relax the rules for a little bit. And before we look at the way that they've done that, and that will be very brief, let's just look at ways around the normal regulation if you do have an ATMP and even a normal medicinal product. So there are alternative routes that will get you a fast track. And what I've done now is I've written down the official reasons and the official ways down these routes, and then below I've written down the simple interpretation, what that actually means. Now, starting on the left here, we've got things where there's actually a lot of data available, and over to the right is less and less data. And everything on the left here applies actually to all medicinal products. And there's one measure that just applies to ATMPs. And equally, everything over here is an EU, and everything over here is responsibility of the country that you're in. So if we start off with the accelerated pathway, this is something that's come up quickly, that suddenly got very interesting, and what the EU is saying is effectively we can save a lot of money and save a lot of lives, so let's just shorten the process. It's obviously very good, high health economic potential. Or it's better than nothing. Or it's something that where there's such an unmet need, there's no other alternative, then what we can do is effectively put this in a program. It says priority medicines program, that again provides advice and shortens the process. And just an example, this was last year, but 11 of the 12 things on the PRIME program were gene technology medicinal products, things like the CAR T cells. Now, that's when there's really actually enough data to say it's good, but you don't want to wait this whole process out. There's data very quickly saying we can turn it into a product. Then there's less data over here. There's less data perhaps because there are too few patients and you expect it's going to take a long time to accrue. Good, then they say we'll use it, then start off and we'll see later, conditional approval. Or there are so few patients you're never going to get enough, and then there are exceptional circumstances where they say we won't just shorten the process, we'll ditch it. You can go ahead and start under these exceptional circumstances. Then we get into a really dodgy area at the end. So on the right we've got very little data. If there's a very, very serious condition, okay, so we're talking about life-threatening condition and no real alternative, this is why I've put nothing to lose here, then we're into the so-called compassionate use program that's really the responsibility of the local authority. And right on the right-hand side is the thing that some people grasp onto, other people hate. It gets used for all sorts of things that it shouldn't be used for. This is the famous hospital exemption. So very little data, very hopeful clinician, very optimistic interpretation, usually, often biologists behind it. Custom made, always locally in the same hospital that it's used in. And these are all local responsibility, exceptional circumstances, hospital exemptions. So the hospital exemption, as an example, are the bone marrow cells into heart. There's no real data there. but there was a hope that it might do something, and it was all in the hands of one surgeon. So the hospital exemption, it must be seen as an exception. Sometimes it's seen these days as a sort of way into the whole system, and based on, at the moment, a lot of different hospital exemptions with similar applications that we can get into an ATMP approval. No, that's not the idea. So it's a way of doing exceptional things. It shouldn't be seen as a way into the system. Okay, the latest thing, and this is worth mentioning because these are the new guidelines that come into operation next week. So this whole process was started 2015, it was adopted at the end of 2017. And the idea here was to say, okay, ATMPs are a problem, what can we do to relieve the situation? There are 80 pages in this document, and I can gladly give it to anybody who wants it, but there are really two main things, I think, that come out. Many small changes, but two main things that come out. The first is the whole organisation is relaxed to a so-called risk-based approach, where the emphasis for calculating the risks and containing them comes away from the regulator and goes to the person who's making the drug. So it goes to the manufacturer, who in this case very often are also the clinicians that are using them. So you put the emphasis on the manufacturer to judge the risks, know what they're doing, know what they're working with, state all this in risk assessments, and then show what they're doing to contain the risk. What the EU and the authorities have to do is to increase their inspection. So they put more inspection duties, and the emphasis for actually making the decisions is pushed down onto the people who are making the products. And they're reschooling the whole of the inspectors now in order to be able to do this. The other thing that really is relaxed is the whole way that the ATMPs go through this series phase 1, 2, 3. Because previously, to get a clinical trials authorization, to get a marketing authorization, you had to have taken the hurdles. And what they're saying now is, well, we'll build the hurdles as we go along. So we will accept that the processes are going to change. We will accept that you're going to be learning about the product you started with, and you're going to know more about it at the end. We will accept that you might not actually see what the mechanism of action is until you've treated a few patients. And we'll put the whole thing on a sliding scale, so you establish these things as you go. It's not that you don't do them, you establish them as you go. So they are trying their best, the EU, and they're trying to relax the conditions. We will see how this is accepted. It certainly puts more work back onto the sponsor and the manufacturer, but certainly it's encouraging that this is a dialogue and that they're moving a little bit to loosen up the field. Okay, right at the end, we heard this morning about MSCs for GVHD. We heard about the Rifkin project. I'd just like to point out to you, the Rifkin project was originally supposed to be 6 manufacturing centres doing all their manufacturing quality control and treating patients. We're 3 years into the project, and we've got 2 established at the moment, because it's proven so difficult to get the different countries working together. And these are how harmonization is. So this is the harmonization issues. This is our problem in the middle. We have some who had loads of paperwork in those cells. We had others who were producing cells without the GMP paperwork. And we have differences not just in the groups, but actually in the national authorities. Some national authorities had a problem with the serum that we were using. This is a Canadian serum. It needed a European qualification. We have others that say you have to include antibiotics when you're collecting bone marrow, otherwise there's a chance of infection. And then the German authority said, no way can you use antibiotics when you're collecting bone marrow, same reason, because there's a chance of infection. And if there is one, you need to know that it's there, so you cannot suppress it. This is an example of how the new guidelines have gone on. They say you can use antibiotics, but they must not affect the sterility testing later. I don't know how that's going to happen. You have to wash them away, I guess, before you do it. All sorts of problems that have taken us a very long time to harmonise. And these are harmonisation issues that are going to be relevant in the different countries. I'm not going to go through that. That was the solution to the quality controls. Okay, so I've gone through this fairly quickly. I'm very happy to talk about specific things with other people afterwards, but I didn't want to take too much time. So I've told you where the ATPs come from and more or less what sort of types there are, how difficult it can be getting an idea into the clinic. This is more something for people who are really developing new products. I'd like you to understand good manufacturing practice, and if you're setting up productions here, then please talk, because this shock picture, everything that was in there, was really just there to say, be aware of the direction it's going in. And I've talked about the special considerations for ATMPs. I have one last slide for Dietka, because we were in Salamanca last week, and I realised Dietka likes roundabouts. So here we are. This is the way that most people have been looking so far at the regulations. So the European Medicines Agency controls or advises national competent authorities. They actually make the decisions and impose on the sponsor and the manufacturers. They impose the regulations on the clinician gives the treatment to the patient. That's okay for some things. For ATMPs, that's not going to get us anywhere. The system that we've got is not good enough. So this is the system that we're actually in at the moment. People talk to each other, and it is up to us also as end users to go back and give this feedback to the regulatory authorities and the EMA. And then you get into a situation where even if you have to go once or twice round before you know where you're going, there's still some way out. And that's where the ATMP field is at the moment. We're looking for the right solutions to the right problems. Okay, thank you.